Hello, my name's Dr. Michael Zimmerman. I'm an associate professor of biology at Shenandoah University, and my specialty areas are parasitology and infectious disease. Now, when we're talking about different types of vaccines, we're talking about different forms of the pathogens that are going to be injected into a host to kind of elicit that initial immune response and create a memory for those different pathogens. And so we can categorize vaccines into six different categories, which we'll go over briefly and give specific examples of before we get into detail about RNA vaccines. The first type of vaccine is what's called a live attenuated virus vaccine. And so obviously there's going to be specific to viral pathogens. And so what this means, the word attenuated means weakened. So what we're doing in a live attenuated virus vaccine is we are injecting live virus into an individual. However, it's severely weakened in that it will not possess the ability to cause disease. And so we're actually injecting live material in here, okay? But no disease is going to be caused from the actual vaccine because the virus is so weakened, it can't perform its normal function and its normal pathological features. Now, the advantage to these live attenuated virus vaccines is that they're very, very powerful and they produce lifelong immunity. And so, 1 to 2 doses of these attenuated vaccines are all you need to protect you for your entire life. So, you get them when you're a young child, then you don't have to worry about getting boosters or getting any additional shots of these vaccines because they institute that lifelong immunity. Now, the reason why these vaccines are so powerful is because you're dealing with live organisms. An immune response is always going to be stronger with a live organism compared to a dead organism. And so you get that lifelong immunity and need very few doses. The disadvantage here is that they're difficult to maintain. Because you have to maintain consistent live cultures and keep generation after generation after generation going, you need to have a very high tech lab setup in order to administer these vaccines. So, somewhere like the United States, where we have all of these medical centers scattered throughout the country, it's a very feasible thing to maintain these live cultures as we need them. But if you're going into more impoverished areas and impoverished countries, they don't have these same facilities. So, these live attenuated vaccines are more common in the more financially and economically able countries like the United States. One example of a live attenuated virus that we oftentimes give to children here in the US is the varicella vaccine or the chickenpox vaccine. You get 1 to 2 doses as a child, you get the weakened virus. It's oftentimes so weak you don't even show the symptoms of the chickenpox. And then you're also protected from shingles down the line due to this weakened strain of the virus. A second type of vaccine is called an inactivated vaccine. And so these are some of the more common vaccines that we tend to administer are inactivated where the virus or the bacteria that we're trying to protect against is actually dead. So you're injecting dead viral material or dead bacteria into a host in order to confer that immunity. So just like the example video that I showed previously, you're going to have that conferred immunity. So your body reacts to that dead bacteria, and then you're going to elicit a memory of an immune response to these dead bacteria. Now, because the bacteria or the virus is dead when it's injected, these are going to require booster shots. So you're going to get another dose, usually a lesser dose of this virus or bacteria later in life to kind of re-trigger that memory of your immune system and kind of reinnervate it and strengthen that immune response since it wasn't as strong as, say, a live attenuated virus in the first place. But a major advantage here with these inactivated vaccines is that they're very easy to maintain. You stick them in a freezer, and then you can give doses whenever you need to give doses because you don't have to worry about maintaining a live culture. So the polio virus is an example of an inactivated vaccine. And so polio virus has been eradicated in the United States for decades because it was easy to distribute this vaccine because you didn't have special conditions to maintain these cultures. It was just recently announced that Africa has also eradicated the polio virus. And so hopefully soon, because of its easy to maintain and easy to distribute tendencies, polio will be be added to smallpox as an eradicated disease worldwide pretty soon. Subunit vaccines. Okay, so subunit vaccines are only going to include antigens of the pathogen. So remember, antigens are going to be external proteins associated with a specific pathogen. And so if there's something on the outside that can be recognized by our immune system, again, it gives it a head start. And so we can just use the subunit or just a piece of that virus or bacteria as a recognition site. And so the only problem here is that you get reduced reaction to vaccine over time. And so as the virus or the bacteria adapt to the immune system, they can potentially change some of those antigens on their coat. And so you oftentimes have to get redosed on these vaccines and boosters and whatnot to update these vaccines. So hepatitis B would be an example of a subunit vaccine. Toxoid vaccines. So oftentimes you have toxins associated with bacteria. And so bacteria produce endotoxins or specific toxins that kind of poison the immune system so they can replicate and cause disease. And so toxoid vaccines are going to be specific to bacteria that produce these toxins. So things like tetanus. And so tetanus is going to be produced by bacteria that produce this toxin that cause your muscles to stay in the contracted form, in the form what we call tetany. And so what happens is these bacteria infect, they're usually associated with rusty surfaces. The bacteria infect the host, they release the toxins, which causes the tetany. Well, the toxoid vaccine is going to recognize those toxins, block the toxins before they can cause tetany, and then your immune system will go in and take care of the bacteria, but no pathology will be caused. Last two types of vaccines, conjugate. Conjugate vaccines are going to target polysaccharides of bacteria, so components on the outside in the cell wall of these bacteria. And so you have very limited scope on these because each one has a unique polysaccharide sequence or polysaccharide coat associated with it. Bacteria flu, so Haemophilus influenzae, is going to be used for the conjugate vaccine, one that's commonly given out, where it targets that cell wall and kills those bacteria. And then the last major category here, and the one we're going to focus on for the rest of this brief lecture, are DNA and RNA vaccines. So DNA and RNA vaccines, you create viral or bacterial genes. So we've actually been able to sequence these pathogens. We know what all of the genes do in those virus or bacteria cells. And so we highlight specific genes that are very, very important or easy to target. And then we allow the host cells to produce the viral or bacteria substances or antigens so that they can elicit their own immune response against it. And so one thing to note about these is that oftentimes these are still in trials. And so one of the first vaccines that targets either DNA or RNA sequences to be piloted is actually the coronavirus vaccine or that COVID-19 vaccine. And so that one has gone through rigorous trials and it's been deemed safe enough to administer to the public against COVID-19.